Okay, so hi everybody. We will start this seminar today, and we are going to talk about the new hidden regulator, the tRNA fragments, their role in cancer and how we can study them. I want to introduce first myself. I'm Alessia and I'm the Innovation and R&D Project Manager in Immagina. Immagina is a small company, as you can see from this picture. We are about 20 people, and our headquarters is in Trento, in the northern part of Italy, in which we have our hub with the innovation and R&D, services, and production. We also have a subsidiary in US, in New York, and a variety of distributors in Asia. The webinar of today is structured in four main parts. First, an introduction on tRNA fragments and how they are generated. Then some challenging on studying this type of molecule and which are the solutions that Immagina is providing. And then we will have a case study on a type of skin cancer using our internal data that we generated, and finally some conclusion and future perspective on this topic. So starting from the introduction, tRNA fragments are generated from the full-length tRNA. So tRNAs are adapter molecules that are necessary for translation, linking the mRNA codon to their corresponding amino acid. And each tRNA is about 76 nucleotides and form a specific cloverleaf 2D structure with acceptor and loop arms. The function and stability of the tRNAs are heavily influenced by the presence of post-transcriptional modification. Even if those are small RNA and maybe small in size, they actually play a huge role in gene expression and in disease development. So tRNA fragments are produced upon nuclease cleavage at the level of the loops of the full-length tRNA. Usually this enzymatic cleavage is triggered by different stress stimuli, and this cleavage will generate a variety of fragments which can participate to various biological processes through different mechanisms of action. But which are the nucleases involved in their generation? So we know that the first enzyme described to produce tRNA fragments was angiogenin, and followed by that discovery, we know that also RNase L and RNase A are involved in the cleavage of the full-length tRNA. All of these nucleases belong to the same superfamily, which is the RNase A superfamily, which also leave a specific cleavage signature, which is represented by the presence of a 3' phosphate on the cleaved RNA molecule. Even though not all the nucleases involved in biogenesis of tRNA are still known, and there is still an active area of research to try to identify other nucleases involved in their biogenesis. However, the tRNA fragments that are produced can be classified according to their length and site of the cleavage, and we can have 5 different classes of tRNA fragments. We have the 5' or 3' tRNA half or tRNA long. Usually these type of fragments are generated from the 5' or 3' counterpart of the full-length tRNA. This fragment has a length of more than 20 nucleotides, and usually the cleavage occurs at the level of the anticodon loop. Then we can also have shorter fragments that are classified in 5' short and 3' short tRNA fragments. Usually these fragments has less than 20 nucleotide in length, and they derive from the 5' or 3' counterpart of the full-length tRNA. And all the tRNA fragments that do not fit any of the categories I was describing before are classified as internal tRNA fragments or also as others. Even if they are fragmented, the tRNA fragments still have a role in translation, as described by many labs. So, for instance, by Ivanov lab, we know that upon oxidative stress, tRNA fragments from alanine and cysteine brings to inhibition of global translation because they drive stress granule formation by sequestering translation initiation factor. By Polacek lab, we also know that tRNA fragments may interact directly with ribosomes by inhibiting global translation or pushing global translation depending on the type of fragments. There are few reports, I think one report, saying that tRNA fragments may inhibit the translation of a specific mRNA in a miRNA-like mode. While for tRNA fragments from leucine was described how these tRNA fragments may bind to the 3' UTR mRNA by unfolding a region within the UTR and enhancing the translation of a ribosomal protein, and so pushing the global translation and ribosome biogenesis. So, summarizing, we have described how tRNA fragments are generated and possible mechanism of action. What we didn't say is that actually tRNA fragments are expressed in all biological models, but the altered expression are already reported in several disease conditions, such as viral infection and neurodegeneration. And today, actually, we will have a focus on cancer specifically. We have to say that the function of these tRNA fragments in disease depends on their sequence, structure, and presence of post-transcriptional modification. So, focusing on cancer, there are already several studies that report specific tRNA fragments alteration, and these alterations are linked to tumor metastases and invasion, to treatment response to therapy, and to tumor growth and proliferation. More importantly, tRNA fragments were also found to be released in bloodstream in complex with proteins or inside extracellular vesicle. So these fragments may be used as a possible biomarker in cancer for diagnosis and prognosis. But I say that fragments are just side degradation product, but they have a biological function and they have a diverse origins, since they may be generated from different tRNA molecule and isotypes. Canonical RNA tRNA fragments may also interact with proteins, expanding their regulatory scope. Their expression is sensitive to stress, development, and disease, and that's why, together with the fact that they can be easily found in body fluid, we can use them as a potential biomarker or also as a therapeutic tool. So, moving forward on how we can study this type of fragments, we have for sure some challenges, because these fragments are deriving from the full-length tRNA, so those fragments are full of post-transcriptional modification as their progenitors, and moreover, as we were mentioning before, the nuclease involved in their cleavage leaves another specific signature, which is the 3' phosphate, which hampers their inclusion in a canonical small RNA-seq library. Moreover, this tRNA has also a high sequence similarity. You can find a continuation from full-length tRNA, and there is a lack of standardized protocol. So in Immagina, we try to solve all of these issues by using two main technology. One is the DORS platform that is intended specifically for tRNA fragments, and I will explain in a moment how this platform works, and the nano-tRNA-seq, which is the nanopore-based approach for full-length tRNA analysis. So what's the DORS platform? DORS maps all the RNA fragmentome, capturing only the 3' phosphoRNA fragments with the zoom on tRNA fragments. It is based on two main proprietary Immagina technology, the tRNA-seq for the omic profile and the 3qPCR for a high throughput screening approach. And these two wet lab approach are complemented with the BioIT pipeline called PROC, which allows for the identification of specific hits. And in particular, with PROC, we are able to have an whole fragmentomic profile, but it also includes a machine learning module for hit selection and in silico prediction of tRNA fragments functions, such as complementarity with mRNA target, presence of post-transcriptional modification, folding, and then once the hits are selected, we can design primers specific for our tRNA fragments of interest for a high throughput screening by qPCR. So just to show you why our tRNA-seq is different from a canonical small RNA-seq, I'm just reporting in here the workflow of this library. So as we can see, it includes only fragments that present a 3' phosphate. And the main features of this library is the direct ligation of these fragments using our proprietary enzyme, the optimized linker design, which enhance the ligation efficiency and minimize the bias. And we also introduce unique molecular identifiers in order to remove PCR duplicates and enable an accurate quantification of our fragments. But just to show you better, this, if we compare our tRNA-seq with the commercially available small RNA-seq library, what we can see is that even though the read length distribution of the two libraries are very similar because they are both capturing small RNAs, what is really different is the content of the RNA that the two libraries are capturing. In fact, with the tRNA-seq, we are enriching in tRNA fragments while not capturing, for instance, microRNA, because this type of small RNA are known to be a 3' OH by biology, and so they cannot be included in our library. So now I will showcase what we got in our internal R&D using our DORS platform for tRNA fragments expression in skin cancer. We have this proof of concept in particular usually remove the lesion at a small percentage of there is a recurrence, and usually recurrence is associated with the poor clinical outcome. Even though in 2018 it was approved a new anti-PD-1 immune therapy, still 50% of the patients are not responding to therapy. So this type of cancer still need a molecular marker for early diagnosis, second lesion prevention, and treatment response. Because nobody look at tRNA fragments in this type of cancer. And so the first very question that we asked ourselves was, is it possible to use tRNA fragments to distinguish between healthy samples and CSCC patients? To answer this question, we use three different methods. The first one are lines derived from healthy donors and CSCC patients. Then we switch to matched skin tissue, in which from the same patients, both the healthy skin and the lesion was taken. And then in the end, we screened the plasma by qPCR of healthy donor and CSCC patients. So starting from the primary cells, we perform our tRNA-seq library. And with PROC, we identify at least 2 clusters of RNA fragments that were able to separate the healthy keratinocyte from the CSCC samples. Among the RNA fragments that were able to clusterize samples, we also found tRNA fragments up and down regulated. Look at the type of fragments that we retrieved. We noticed that most of the upregulated tRNA fragments were belonging to the internal class or to the 5'half class, while the majority of downregulated tRNA fragments were belonging to the 5' long or 5'half tRNA. Using feature and very nice between keratinocytes and CSCC. And by we remove the tissue, our RNA-seq in skin tissue, and again, even if less nicely than before, we were able to identify 2 clusters of RNA fragments that distinguish between normal skin and CSCC lesion. And again, we found numbers of significantly up and down regulated tRNA fragments. From the PCA, we still were able to distinguish the 2 classes of normal skin and CSCC, even though, again, less nicely due to the nature of the samples itself. But however, we combined the 2 data sets between the primary cells and the skin tissue to see if there was some tRNA fragments that was in common. And the answer was yes. We identified 100 tRNA fragments that were in common, and some of them showed an heterodirectionality, being upregulated in skin biopsies and downregulated in primary cells and vice versa, while others showed an homodirectionality, being upregulated in both primary cells and skin tissue, and vice versa. So we move forward and we look at the cleavage pattern of these in common tRNA fragments. And what we found is that 4 tRNA fragments between the 100 exhibited an identical cleavage pattern at the nucleotide resolution between the 2 datasets. These fragments were derived from the glutamate, alanine, glycine, and aspartate, and 3 of them showed a homodirectionality, with the glutamate and alanine being downregulated in both datasets, while glycine was upregulated, and the aspartate was the only one showing an heterodirectionality, being upregulated in cell and downregulated in the skin tissue dataset. Once we identify these 4 tRF, we wonder whether these fragments were also found in the plasma samples of patients, since we know from literature that tRNA fragments can be released in the bloodstream. And so we decided to screen several plasma samples coming from patients and healthy donors for the specific fragments that we identified. And as we can see from here, the different fragments shows different features. So we found that the glutamate was already significantly downregulated at the very early stage of disease, and this downregulation was maintained with disease progression, while for the alanine, the downregulation was significant only at a specific stage of the disease. While for the glycine, after an initial significant downregulation at stage 1, it seems to have a trend toward upregulation once the disease is progressing. So overall, we may say that with tRNA-seq and 3pQPCR, we enable a deeper exploration of the biological and clinical relevance of these tRNA fragments. And with this, I want to go on the overall conclusion. So I hope that the study of tRNA fragments serve as a striking example of biological complexity, where the fragmentation of the tRNAs has given rise to a new world of structural and functional diversity. So by combining data that comes from structure, sequence, and cleavage pattern of this molecule, we can gain a deeper insight into their role, opening new venue for biomarker and therapeutic target discovery. Overall, we can say that tRNA fragments may be integrated at different stages of biomarker and drug discovery pipeline, from the preclinical phase, it is actually already used from some companies that are developing tRNA-like drug, but they can also serve as a predictive biomarker for all those diseases that are lacking reliable molecular signatures. I want on the fact that internally in Immagina, we do not cover cancer today, but we also internally develop a pipeline for spinal muscular atrophy, which is a rare neurodegenerative disease, and we are starting 2 new cases, which is a metabolic, and on inflammatory bowel. Overall, I want to say that this is just part by Immagina, which aims to study the whole translatome, and some of the technologies, such as for tRNA, can be complementary to our DORS platform. Look at present modification on the full-length tRNA. Now the tRNA is then influenced by this modification. And this I want to conclude, and thank you, and answer your question, if any.
I think someone raised their hand to speak. Kuldeep, I think. Let me see. Go ahead, feel free to ask questions.
Yes. So first thing I would just suggest, because we also work on tRNA fragmentation. In the first slide, when you showed the angiogelin is the ribonuclease which cleaves the tRNA, but you did not include IRE1 because there are multiple studies which shows the IRE1 also involved in tRNA cleavage and tRNA fragmentation. So that is one thing, but I also wanted, okay, that will be different, but I also wanted to know what are the cost of these services if someone has to use from Immagina?
Which other services?
Yes, like if we have to do these sequencings, or we have to look for tRNA, if some lab wants to use this facility, how much will be the cost? Or
Okay, so for the pricing part, let's say that I will leave it to our sales representative. I'm more on the R&D part. So if you're interested, I can put you in contact with our sales. Are you working on a specific disease?
So it's with you. I'm just wondering whether someone is coming to moment.
Okay,
Just in case.
hello. Hello.
Hi.
Yes, we can hear you. I
Sorry,
haven't heard Alessia's question.
I was in lab, so yes, that would be great if you can connect me with some representative whom I can ask about the services.
Sure. Yeah, sure. I can put you in contact with one of our sales representative. I was just wondering if you are working on a specific type of disease or.
We work on cancer, but what we also discovered something like if you know the Kiosin modification.
Yeah.
And the Kiosin modified tRNA, they are fragmented when they are modified with PQ1. And this is our paper, which is accepted in mold cell and cell biology. So what we found interesting that the same tRNA which are modified with the Kiosin, if they are modified with PQ1, they are cleaved. And so we are working on it. So in that context, I wanted to.
Okay, so sorry, from which lab are you?
I am in Montpellier, France.
Okay. Okay. Thank you. I will put you in call.
Sorry, I think this glitched out there. Can you repeat what you just said?
So, okay, I think that there is another question. Thank you for the talk. Is the chemistry compatible with the small RNA with cyclic phosphate at the 3' end? Definitely yes, because the enzyme is taking both the 3' phosphate and the cyclic phosphate. So it's compatible with both.
Okay, because what we found, we have to cleave with this PNK cyclic phosphatase before adding the adapter.
Okay.
If they are cleaved by, if you are looking for IRE1 specific cleavage, you have to use the enzyme, the PNK cyclic phosphatase before the adapter adding.
Okay. Yeah. Yeah, I confirm that is compatible.
Okay. Thanks.
Yeah.
Okay, I think that was all the answers, all the questions we had. So if there's no further questions, I think we can end the webinar here, right?
Yeah, I think so.
Nice. I think, yeah, most people have left, but in any case, thank you everyone for attending. And yeah, then we can stop the webinar. This webinar will be made available on our YouTube channel afterwards for those that didn't attend. And even if you want to rewatch the webinar, you can do so there.
Okay, thank you everyone. Bye.
Okay, thank you. Bye-bye.